My name is Bill. I am the lead for the GDC User Services team. I am joined by my colleague Lauren, who is a senior bioinformatician for the Genomic Data Commons. And today we would like to talk to you about single-cell RNA-Seq support in the GDC. Um, Sorry, I pressed the wrong button too. Here we go. So I just want to go over a short agenda just to show you what this presentation will be about. First, there's going to be a very short introduction about the GDC and single-cell RNA-seq in general. Next, we're going to talk about single-cell RNA-seq as it exists in the GDC. Then Lauren's going to get a bit more in-depth on how the GDC processes single-cell RNA-seq. That'll be followed by a demo given by Lauren with a Jupyter notebook on how to visualize single-cell RNA-seq data. Then finally, there will be some announcements and we'll take any questions. Just to remind everyone, you will remain muted throughout the duration of this webinar. At the end, we will answer any questions. Please, during the entire webinar, you can type your questions into the Q&A panel. There's like a chat panel too. Use the Q&A panel because that's where we're going to look and we don't want to miss any questions. So now starting with an introduction. For those of you who might be new to the Genomic Data Commons, here is a diagram of the GDC harmonization flow. You might have seen this in previous GDC webinars, but the flow basically goes that the GDC submitter provides raw molecular data. This can be a BAM file, it might be already aligned, or a FASTQ file. It doesn't matter. It all turns back to FASTQ during alignment. Then we align it to the reference genomes, GRCH38, which, if you're interested, can be found on our reference page in the GDC website. The aligned reads are made available in BAM format, and we also perform downstream analyses based on experimental strategy in addition to the BAM. So as you know, if there's RNA-Seq, we'll be doing expression quantification. If it's whole genome, whole exome, we might have some variant calling. And all the alignments and the downstream analyses are all made available in the GDC data portal, which is where the data is downloadable. So now that I've introduced the GDC a little bit, I want to get into single-cell RNA-seq. This is just a very basic diagram, and I'm sure most of you are bioinformaticians and are at least familiar with RNA-seq. Starting with the step that is RNA right here, that is converted to cDNA for stability, then amplified, put it into a sequence library, and then sequenced. That's the basic workflow for RNA-seq. With single-cell RNA-seq, there's an extra step there. And the extra step is isolating single cells so that they can be processed separately. This is done in many ways. Some sort of flow cytometry or some other type of separation happens. And each cell is barcoded so that later on, each single cell can be differentiated from one another in the sequence file. The RNA-seq happens along with the barcodes so that you can tell them apart. Each expression profile is performed on each single cell, and transcription profiles can be identified based on one cell rather than traditional RNA-seq, which kind of happens across many cells all at once, all in the same mix. So now that short introduction's done, I'm going to tell you a little bit about how single-cell RNA-seq can be retrieved, queried, etc. in the GDC. So in general, the GDC processes single-cell RNA-seq using these pipelines. First is Cell Ranger, which performs the alignment and the gene expression analysis. And then that's followed by Seurat, which performs the secondary gene expression analysis. It's an R toolkit. I'm going to get into a little bit more about what that needs, but that's just the basic workflow. Cell Ranger and then Seurat. So here's a diagram that I put together. We get the two FASTQs in. Cell Ranger aligns them, so that alignment is made available in the GDC. And it produces two gene expression matrices. These are basically what the expression level is for each gene for each cell. This is made available as a raw count matrix, which has all of the barcodes in it, and the filtered count matrix, which only has the cellular barcodes, the barcodes which were determined to be associated with a cell. There could be some other barcodes that are there. They could be associated with free-floating RNA or a cell that was lysed and maybe attached to two barcodes. But the filtered count matrix is the one that's safest to say is associated with actual single cells. This data is then processed with Seurat. So remember, this is the regular gene expression data. And Seurat will do clustering and differential gene expression analysis on this data. And I'm going to get a little bit more into how that is displayed within the files in the GDC in the next upcoming slides. So just to list them all out without too much of a workflow here, I just want to let you know that all GDC analysis is on the GRCH38 reference genome. And as of now, September 27th, 2021, it's GenCode version 22. You're going to see an announcement in a later slide that will detail our migration to version 36 soon. But for now, version 22. The single-cell RNA-seq data has 5 data types and 6 major output files for each sample. So there is one BAM file, the 2 gene expression files, which are available as gene expression quantification data type. There is one analysis file that is the single-cell analysis. That's where you get the clustering. One differential gene expression file, that's the differential gene expression, and one full output file, which can be found under single-cell analysis. I've color-coded them. As you know, if you've used the GDC before, GDC data is available as either open access data or controlled access data. And in the single-cell RNA-seq experimental strategy, only the BAM files are made controlled access. These require dbGaP access. The rest of them are completely open, can be downloaded by anyone without logging in or starting an account or anything. You could download them right now. So, in terms of availability, I'm going to give a really quick demo on how to reach this data. So this is portal.gdc.cancer.gov. It is our data portal. If you go to this website, you would click on repository, go down to files, and go down to the experimental strategy. Because there's not that much single-cell RNA-seq data, it's going to be towards the end because it's arranged by the number of files. So,
Bill, we can't see your screen.
You can't see my screen. I'm sorry.
No, that's all right.
Let me go ahead and reshare that. My apologies. I must have pressed the wrong button to stop sharing. Okay. Let me get this Webex out of the way. Okay. Sorry. I will start over. So we're in the. You start out at the data portal. You would go to the repository and scroll down to experimental strategy. Because single-cell RNA-seq has the fewest number of files right now, you'll have to click the more button at the bottom. And then when you search for that, it'll separate it down by workflow type, data format, and data type. And the data type is what I outlined in the previous um, slide. So for example, if you wanted to look at single cell analysis, you can click on this and download either the analysis files or the HDF5 Loom files. So I will go back to the PowerPoint. Here we go. So as of today, single cell RNA-Seq is available in the GDC for the CPTech-3 project. This is a project that uses that has a lot of sequencing genomic data and also has a lot of proteomic data, and the purpose is to associate the two. Um, the single cell RNA-seq files are available for 18 cases. All of these are from glioblastoma patients. All samples originate from primary tumor, and all of these samples are actually single nuclei RNA-seq, which is a different type of single cell RNA-seq. And in this case, RNA was extracted from the nucleus rather than from the cellular cytoplasm. So um, that should be processed. Oops, sorry. That should be processed accordingly. Um, One more thing I want to point out is that if I just showed you before, the single cell RNA-seq can be reached by going to experimental strategy, single cell RNA-seq. But if in the future there might be more projects that might not be single nuclei RNA-seq, and that can be filtered using the property aliquots.analyte type, and you can reach that by going to add a case biospecimen filter on the top and selecting nuclei RNA. If you're wondering which single cell library the sample came from, you can determine that with this property, readgroups.single-cell-library, and you would reach that by going to add a file filter. Right now, it's only nuclei RNA and the chromium 3' gene expression library, but in the future, there may be other projects. So this might be a bit more useful then. Now to get onto the file formats. So as I said before, the gene expression files are available as filtered or raw. Um, they are the the gene expression for each gene for each cell. So this is actually made available in the market exchange format, which is a tarball. Once you unzip that tarball, you will see three TSVs that are actually gzipped. The barcodes, of course, are a list of the barcodes. The features are the list of the gene ID and the gene name. And the matrix is the actual gene expression matrix. One thing I do want to point out from some help desk queries is that if you're using a Mac and you have these three gzip files in your browser, they may not be able to unzip by double clicking them. So in that case, you would go to the terminal and just run this command, gunzip barcodes.tsv.gz, or whichever one of these you're looking to unzip. So just want to make all users aware of that. Next is the analysis file. This gives you the information on how to visualize and where the clusters fall based on their transcription profile. So this includes things like the cell barcode, the read count, the gene count, the cluster that it falls into, based on what Surratt said, and then coordinates so that users can generate either a UMAP, t-SNE, or a PCA plot based on just match up the columns based on the column name, and those that is how the data can be visualized. So next I want to talk about the differential gene expression. Um, this contains information on the differential gene expression between clusters for differentially expressed genes. So each line includes which cluster is being compared to the rest, which gene we are talking about, the average in fold change, um, the p-value, the adjusted p-value, the proportion of cells in the cluster, and the proportion of cells outside of the cluster. Next, we have available the full Seurat analysis log. This contains the full analysis from Seurat. It's available as a Loom file in the HDF5 format. And the purpose of this is to store the entire analysis on disk rather than in memory, because it can get very large because of the number of cells that are being processed and the number of genes. So when you multiply them, you get a very, very large analysis. There are several R or Python packages. The ones that were recommended on the Seurat website were LoomR and LoomPy. And next, I will turn things over to Lauren to talk a little bit more about the single-cell RNA-Seq harmonization pipeline.
Thank you, Bill. So I am going to talk a little bit more in depth about the single-cell RNA-Seq harmonization pipeline that's processing all of this data. So first, we have the 10x Genomics Cell Ranger tool that was developed by 10x Genomics. And the input are FASTQ files that have been generated using 10x Genomics technology, and it performs alignment, filtering, barcode counting, and UMI counting. And as Bill said, there are about 5 data types that come out of this and are available. And the ones that we use downstream are the filtered feature barcode matrix file, which contains only cellular barcodes in the MEX format. And yeah, so those are used for further downstream analysis. Everybody is, let me do it like this. Is this better? For, so then we have the H10 single cell RNA-Seq QC and filtering workflow. This was developed by Josh Campbell's group, and he developed this tool to further QC single cell RNA-Seq analysis using R. And this takes in the filtered matrix files from Cell Ranger. It could also take in files from other single cell RNA sequencing workflows like Star Solo and others. So what this does is it collects a bunch of QC metrics on the data, so contamination, doublets, empty cells, etc. And you can see here in this table the different methods, R methods, and packages that are used within this this tool. And based on thresholds that are defined within these tools, we have a score, and if cells are outside or outliers, or are determined to have either doublets or some major contamination, we exclude them further from the data set. And so the input from here are the raw and filtered MEX files, and then the output is a newly filtered MEX file with an annotated barcode file indicating which cells have been filtered out based on this method. And And then we continue on to this Seurat analysis using R. And this is to identify and interpret sources of heterogeneity from single-cell transcriptomic measurements and integrate diverse types of single-cell data. And then this also takes in either the filtered matrix file from CellRanger or the filtered MEX file from the HTAN workflow that is just a further filtering step. And then the output here is like what Bill said, the differential expression and Seurat analysis, clustering, etc. So now I'm going to show, a small short demo on the Jupyter notebook that we have developed that should be made available soon, I would say, in the next few weeks or so. So first, we have instructions on how to install and download this Jupyter notebook. I'm not going to go in depth into this, but it supports whether you're using RBase, RStudio, or Anaconda. So you can see there's some pretty detailed instructions on how to install and get started. But I am going to talk more about once that is done and you're within the Jupyter notebook itself. This is just a guided GDC single-cell RNA-Seq analysis tool. It is to provide a baseline analysis of the single-cell data of the user's choosing, and provides a stepping stone to further customized analysis that anybody would want to do on their data set of choice. So in this notebook, we will demonstrate two independent analysis with single-cell RNA-Seq. Gene expression visualization on the standard Seurat analysis, and a multi-sample analysis where we integrate the analysis of two samples that were combined. And the input of these analysis these analysis tools are the single-cell RNA filtered count matrices from that are available in the GDC. And both examples are from the CPTech3 project that are available right now. And the only thing you really need from the GDC are the UUIDs from that are provided. And here you also have hot links to the specific example files. So it provides a little bit of a clear analysis and what exactly is needed to start this this analysis. And again, you can find more of these files within the GDC, either and exactly how Bill demoed it earlier in this talk. So to prepare, you need to install some packages as well as a supplementary function, which basically connects to the GDC and downloads the necessary files for analysis. So first we're going to do a single sample gene expression visualization, and we will read one single sample of the single cell RNA-Seq data, and within that single sample there are many cells. And to perform this standard clustering, we will use Seurat. And we will also generate some visualizations on the expression and some violin plots. So the first thing we need to do is read in the data. So we pull this UUID from the GDC, and the sample name can be either the sample name from GDC, or you can put in your own sample name. So this can be sample one, or can whatever you choose. So this is a variable that can be altered, but the GDC UUID cannot be changed unless you are changing it from one sample to another. So once that's read in, you can see that the output is telling you how many unique cells have been read from this sample. So it's about 10,000. And then we move on to the cell clustering and standard Seurat analysis. And this step might take a little bit of time, so I'm not running through this in real time because it takes several minutes, but we do have print statements indicating that the analysis is running, and then when the run is completed. You can also note that a function is running when there's a star in this input bracket, and once it's finished running, it'll have this number there. And then from there, we can plot all of the cell clusters that have been provided by the Seurat analysis in this UMAP plot. You can see there's several different clusters of cells in this plot. You can also adjust this. You can adjust the dimensions of this plot. So if you want to make this bigger, oops, you can make it bigger, and you can add a legend if you would like, but I can just, so if you want, you can make this bigger so you can see a little bit more detailed clustering plot. And then from there, you can visualize the expression of known genes. So for example, right now we have visualization of TP53 and BRAF within this glioblastoma sample. And you can see the expression of TP53 is pretty sparse, but then you have a lot of global expression with BRAF. So it's kind of expressed throughout each cluster, it seems, or at least highly expressed throughout each cluster, which is kind of makes sense for some cancer samples. But then also you can add to this. So if you want to add EGFR, and then we'll make this a little bigger so we can see it. You can add EGFR to this analysis. And so yeah, you can see that we've added that gene to this expression view. And again, there's, it seems like there's not a lot of overexpression of EGFR in this sample. But again, you can adjust this in any way you choose, and also put in whatever gene you would like. You can add, subtract, and customize this as you wish. And then to visualize the top differentially expressed genes, we run that analysis showing or finding these differentially expressed genes and comparing them to all other genes. And then we also put in this seed of 1, 2, 3, 4, 5 to make sure and maintain reproducibility. So for this example, we are filtering the top 2 genes in clusters 1, 2, and 3. This can also be adjusted as the user chooses. So you can, in here, this is clusters. So this variable can be 1 through however many clusters there are in the sample, so about 15. So you can adjust this to 2 and 3, 5 through 8, etc. So that can be adjusted. And also the top genes can be, a number of genes can be adjusted as well. You can choose as many as you'd like. Usually 10, up to 10 is typically what is visualized. But again, this is all kind of a demonstration and a starting point to an analysis that can be very customized to your needs. So here is a table of the top 2 genes in clusters 1, 2, and 3. And we can see here that we can also visualize the expression of these 6 genes. And we can see that each one is more highly expressed in different clusters. So you can see here, FMN1 is more highly expressed in this cluster. It's not really present in the other clusters. And then MTCO1 also very highly expressed here, and kind of at the edge of this cluster. So again, you can see how these top genes are differentially expressed and in what clusters they're differentially expressed. And these might also lead to further analysis in cell type, etc. So again, very customizable, and this is a starting point. We can also inspect the expression with violin plots. So these are the same 6 genes, top 2 genes in clusters 1, 2, and 3. And so you can see here the x-axis is cluster, so 0 through 11. And then the y-axis is expression level. So you can see comparatively how differentially expressed these genes are relative to other clusters. So yeah, that's what this plot will provide. And again, customizable. These can be genes of your choosing as well. And then moving on to the second part of this notebook, we have a multi-sample analysis. So here we demonstrate how to integrate 2 samples for analysis. We can also do this for 3 or more samples, and it can be integrated in a similar way. But for now, I will be demonstrating the integration of 2 samples. We also use Seurat to perform this integration and sample analysis. So again, we have to read in the data, and we read in 2 barcode files, and these again are the filtered MEX files from the GDC. So we have this function again, reads RNA sequencing from GDC, and we provide 2 UUIDs that are available on the GDC page. And you can see each one has a different number of unique cells within the sample. So there are about 10,000 in sample 1 and about 6,700 in sample 2. And then from there, you need to integrate this data. So it's important that these have assigned cell IDs to distinguish which cells came from which barcodes and from which samples. And we also need to anchor that integration. Otherwise, there will be some sort of warning or error. So again, we name sample 1 assay 1. We can name sample 2 assay 2. Again, these can be changed as you choose, whatever you would like to call this analysis. And then we move on to some data normalization that is a standard normalization for this method. And then we find anchor cells to integrate this data. So here we just provide, and again, this is a very slow step, so I am not doing a live demonstration of this, but we do have indicators that show the function is running and when it is finished. And then we can integrate that data together and perform that standard Seurat analysis that we had previously in the single sample analysis. So we also want to make sure we change the default assay to integrated analysis, just to make sure that we aren't overriding or doing anything with the raw RNA that was read in initially. So again, we run this standard analysis on each of these samples, and the same clustering analysis. And we can do some data inspection to make sure each sample has the right number of cells that we originally started with. And then from there, we plot these samples in different ways. So first we can overlay sample 1 and sample 2 clustering together. So you can see they are slightly different as far as their clustering. And then we can also see how each cluster clusters together here. So this is all of the data combined, and this is sample 1 and sample 2 overlaid with one another. And you can kind of look and compare one to the other. So this would be, it looks like cluster 5. This is cluster 4. So you can see how many cells in sample 2 versus sample 1 are within each cluster. And next you can look at a side-by-side view of these clusters. So here is an example of the clustering. So 0 through 13, you can compare them side by side. So this would be, this looks like cluster 4. So this sample has a lot more cells in this cluster than there would be here. And yeah, so you can do that analysis and kind of jump to the next steps once you see that and find differences or similarities, et cetera, within the two samples. So that is what's available as far as analysis in this Jupyter notebook. We also have a session info to show what packages were loaded in this space, and also which versions of R, et cetera, that were being used. Gonna stop sharing here and then move back here. Okay. And then I will give it back to Bill to talk about the data release 30.
Thank you, Lauren. That's the end of the single-cell RNA-seq content. I just want to make a few announcements about the Genomic Data Commons and some current and upcoming things going on. First, data release 30 happened last Thursday, if you weren't aware. And I want to plug a couple of cool new projects that we have. First is the RABC Thyroid project. Both of these projects are are a study on the Chernobyl accident that happened back in 1986. The REBC Thyroid Project uh, takes a look at the thyroid cancer incidences, which was higher after the accident, um, in people who were exposed to the actual radiation. Then we have this project Trio Crew, new in the GDC. This is actually the GDC's first non-cancer study. Every other study in the GDC at this time is a cancer genomic study. This is actually a germline study in which trios, mother, father, offspring, are were studied to determine which, if the parents were exposed to the Chernobyl accident, the radiation from this accident, what are the effects on the mutations of their offspring? So this is data that has some with only the mother, some with only the father exposed, and some with both exposed. And the child was not directly exposed, but they wanted to see what the effects on the offspring were. So data for these two projects are now released to the portal. We also have protein expression RPPA data released for the TCGA projects. Methylation data from the Sesame pipeline is now released for the CPTAC3 and HCMI CMDC projects. We have new cases released for the HCMI CMDC project, and this was released last Thursday. I want to let you know that if you didn't get a notification about this and you're interested in future GDC releases, I would encourage you to join the GDC listserv. Um, I'm not sharing the screen right now, but you can go to that by going to gdc.cancer.gov, going to the support tab, and just click on join the GDC user listserv. It's very easy. From there, you just enter your email and your name. Next slide. In the future, data release 31 will be happening, and the main feature of this release will be that the TCGA slide images will be returning. As you know, they were taken off the portal relatively recently, a few months back, and we're projected to release this in October 2021. Next slide. And this will technically be data release 32, but I want to inform users about this. Um, as I mentioned before, the GDC uses GenCode version 22 in all of its in all of our analyses, and the GDC is now moving from GenCode version 22 to GenCode version 36. What this will mean is that if you look on the portal, all of the files, and when I say V22 files, I mean files that depend on the GenCode. There are some files like whole genome sequencing alignments that do not matter because they are processed completely isolated from the completely independent from the gene model. But the ones that are affected will be replaced on the portal with the version 36 files. All of the version 22 version files that are on the portal will remain available with their original UUIDs. You won't see them on the portal, but if you happen to have a manifest or if you need to query them, you will be able to do so. And I do want to let you know that no new files will be released for version 22. So we do encourage users to download the version 36 data and switch over if you're anticipating using files in the future. This is for if you are involved in an analysis working group. I would encourage you to reach out to the GDC if you haven't heard instructions on how this will affect your files. But this is just kind of a layout for what will happen for conventional users. Next slide. Here are just some references for the 10x Genomics and Seurat. Next slide. And here I've put a few links to things like the portal, the documentation. We have on our documentation site an outline of the single-cell RNA-Seq. So if you want a very short overview of how that works, feel free to go there. If you're looking for the website and webinars, you can go to these links. This recording will appear on the GDC webinars link sometime in the future once captions and editing is processed. And if any questions you have aren't answered here, or if you don't think of them until later, feel free to contact the GDC Help Desk at this support email address right here. You can also find that by going to gdc.cancer.gov. And we're happy to answer questions about anything in the GDC. That's it. You'll receive a response within 24 hours, and someone will uh, address your concern. That is it. I think it's time to take some questions. And thank you all for listening.
Okay, great. Thank you, Bill. Thank you, Lauren. We have a few questions. The first question is there a way now or in the future to download for each sample the FASTQ files in order to perform, again, the mapping process with a different mapping tool?
So that's a good question. So the GDC doesn't actually provide FASTQ files. We only provide aligned reads files. However, anything that's unaligned or aligned with lower quality is still kept in the alignment. So the original FASTQs can be regenerated from the aligned reads file from the portal. So in short, FASTQs will not be available, but they can be generated from the BEM file.
Okay, thank you, Bill. Where is this notebook available? Is there a link?
So the notebook is not available to end users yet. We just wanted to demonstrate it on the webinar. The notebook will be made available to end users in the near future, in the coming weeks.
Okay. And will you annotate clusters generated by Seurat?
Lauren, I'll turn that one over to you.
Yeah. At this point, we were not provided with if that, I think this is what you're talking about as far as like cell type annotation. We did not have that available, but I think that can definitely be available in the future. I hope that was.
Okay, thank you, Lauren. I do not see any more questions. Let's just give it a minute or so to allow folks to type if they are. So we'll just hold on.
I see one more question in the where can we access the new GenCode file? This new GenCode file will be made available on the website gdc.cancer.gov. You can get there by going to the About the Data tab. It's on the main page, and then clicking on GDC reference files. And the GenCode 36 files and the old GenCode 22 files will be found, can be found there.
Okay, and I see a question in the chat. Is there any guidelines for file names?
Okay, yeah, that's a good question. A lot of the file names are actually the same across many different files. We would encourage you to sort them by their unique ID, which can be downloaded along with them. The file names are just named according to what file type they are. So the analysis file has a certain structure. The differential gene expression file has a certain structure. If you click around the data types, you will see that the names tend to follow a certain convention.
Okay, in the Q&A, we have a question. It seems you are using Seurat version 3 functions for integration. Can you explain why you didn't prefer SC transform integration for processing multiple data sets?
At this point, we wanted to keep a Seurat notebook available, so we were using the Seurat v3 functions. So as far as like a streamlined approach for this type of analysis. However, I think it could be possible to also use the SC transform integration for processing, and we can also kind of troubleshoot that and see if we can provide that as well.
And I think so there's some questions in the Q&A. Is the time of day when individual samples were collected available on the GDC website?
No, we do not actually have that metadata at this time.
Okay, and which tools do you use to cell type annotation? Anyone can do the process automatically?
Usually that is provided with the samples that have been analyzed, but there might be ways to do some like marker identification. So if there's a cell type marker that you know of that could indicate which cell type there is, that specific cluster is, that's another way you can do that, but it might take a little bit of a lift.
Okay. I am working with single cell spatial analysis using SERRAT. My question is, when should we normalize our data?
That is a good question. I am not 100% familiar with that, but but I can get back to you on that once I kind of look into that type of analysis.
As a follow-up, and what method
Yep.
should we use for normalization?
That's also a very good question. I would have to look into this a little bit more closely. But if you want, you can submit a question to the help. I forget what that's.
You can submit a question to the help desk, and
yeah,
we'll definitely get back to you.
Mm-hmm.
Hey, is any single cell seq dataset available for splicing transcript variants?
Not at the moment, but maybe in the future. I don't think Cell Ranger provides that information at the moment, so we do not.
Yeah, anything that we process in the near future will be done with Cell Ranger and SERRAT, since that is the current, single cell RNA pipeline that the GDC uses. But in the future, there may be additional data sets, additional pipelines. But for now, this is what we have.
Yeah, and Cell Ranger is constantly updating. So in a few versions, there might be something like that provided as well.
Okay. Are there any other questions? All right. Well, thank you all for attending today's webinar, and we'll try to make the video available for, on the GDC website as soon as we complete processing. Thank you so much, and thanks to our speakers, Bill and Lauren. You all have a great rest of the day.
Thanks everyone for joining.
Thank you. Have a good day.